Hi, everybody. It's Lauren Byrne from the HD Youth Organisation, and we are together again for another Breaking Down Barriers video. As some of you know, this is a series that the HDYO have been putting together to really try and break down key concepts in research and drug development and everything kind of about moving things forward and the barriers that impact people impacted by HD. Today, I am pleased to be with my co-star, our partner in crime, Ashley Clark.
Hi everybody, it's so nice to be here again. Very much looking forward to deep diving into another video.
And I'm very pleased to introduce David Reynolds, who's from LoQus23. And David, if you want to introduce yourself.
Hello everybody, I'm David. I'm really looking forward to talking to you all.
Brilliant. So a good place we like to start, David, is give people an idea of how you and your company got into Huntington's disease and into the community and your experience. I know some facts about you that are interesting, so why don't you give us kind of how you got here?
Thank you, Lauren. So I'm the CEO at LoQus23, but although I'm the CEO, I very much think of myself as a scientist. And actually, my kind of journey with Huntington's disease started when I chose my PhD back in the early 90s now, so quite a while ago. And actually, I chose a PhD on Huntington's disease because I wanted to do research on not just a kind of abstract piece of biology, but actually biology related to a disease, so that hopefully, what I learned during that PhD could be useful to the field and ultimately useful for patients and treating the disease. Now, obviously, the PhD was a long time ago, but I learned a huge amount about Huntington's disease. But to my shame, actually, whilst doing my PhD, I never actually kind of got out of the lab and engaged with patients and with families to really understand the disease from kind of the human side. But be that as it may, that's what started my Huntington's journey. And actually, when I finished my PhD, I decided I wanted to kind of carry on that vein of doing work that's related to finding medicines for diseases. So I went straight into the pharmaceutical industry and spent nearly 20 years working for big pharmaceutical companies, Merck Inc., based in the US, a Danish drug company called Lundbeck, and then another US drug company called Pfizer. And in that time, I worked on many different neuroscience diseases and disorders, not actually Huntington's disease, but I worked on anxiety, depression, schizophrenia, Alzheimer's, Parkinson's. So I really kind of learnt my chops on how to do drug discovery, how to understand these diseases, and then how to pick out the biology that we think could actually be leveraged to find a medicine for people. After 20 years in large pharma, I decided to take a change of tact, and actually I work for a UK charity called Alzheimer's Research UK, which does kind of what it says on the tin, and it funds research into Alzheimer's disease and other dementing, so diseases that affect cognition and function in patients. And when I was there, I was the chief scientist, so my core job was to actually direct the science that the charity funded. But a key part of it was also getting out and meeting people, hopefully being able to communicate clearly science and complicated science to people who aren't scientists. And the other thing I learned how to do was raise money, because charities can't put money into research unless somebody gives you that money. So I spent a lot of time with high net wealth people, so rich people, trying to persuade them to give us money to put that into research. And that was really useful training for then going on to be the CEO at a biotech, where you have to talk to investors and that you want them to give you some money so that you're able to turn your scientific ideas into reality and ultimately a medicine for patients. So that's
Amazing
kind of my
journey.
history.
Yeah, that's great. Thanks for sharing. So I think this is the first time some people in our community might be hearing about LoQus23. So maybe you could tell us a bit more about the company and how it all began.
Absolutely. So I think the key person I also want to kind of introduce at LoQus23 is Caroline Ben, who some of you may know. She is just an amazing scientist, also an amazing
On
human being.
person, yeah, the most
She's
amazing
just
human
an
being.
amazing human being. So
Yeah.
Caroline spent many years in academia working on Huntington's disease. So actually, I first met Caroline when I was a PhD student. We were at a conference
Wow.
in Cambridge, and actually our PhD supervisors, so hers was Jill Bates, who's done an awful lot of Huntington's disease transgenic mouse models, and
Yeah,
I feel
she made the first
really.
Huntington's disease mouse and was part of the people that that discovered the gene or went to Venezuela to discover the gene.
Yes, so Caroline was working doing her PhD with Jill. I was doing my PhD with Jenny Morton, another Huntington's researcher based in Cambridge, and they started collaborating. So actually, my successor PhD student worked on Jill's mice to first start to understand those nearly 30 years ago now. So that's where I first met Caroline. But actually, the story for LoQus23 starts about 6, 7 years ago, where the human genetic studies that Huntington's patients and gene carriers had contributed to had helped us understand that certain biological pathways, which we'll come on to a bit later about DNA mismatch repair, were critically important in driving the disease. So Caroline obviously instantly recognized this was a whole new opportunity for trying to treat Huntington's, related in some ways to those therapies that are targeting the Huntington protein, but a very distinct way that we thought had real potential for coming up with a disease-modifying medicine, so something that can actually slow down or stop the progression of Huntington's disease. So Caroline then engaged with a venture capital group, so a group that can put money into a biotech company to make the science happen. And they actually employed her to work up a business plan of what would LoQus23 do and how would it go about doing it. Caroline is a great scientist, but she didn't want to be leading the business bits of the company. So I was brought in to kind of do the corporate side, as it were. Caroline leads the science, and then we've obviously recruited a team around us to do all the other bits and pieces. So LoQus23 started, it was incorporated on the 29th of March 2019 is when we got going. And it really got going that summer.
Difficult time.
It was. Well, it was before COVID. So we got going, and then, of course, COVID hit. So it did make it quite tricky. The office I'm sitting in today, we got the keys for 3 days before the first lockdown. So I got the keys, I went home, and I didn't go back to it for 3 months.
Oh, wow.
But we did manage to keep the science going through that. So LoQus23 focuses on this DNA mismatch repair biology, which we can talk about. And what we're trying to do is produce what are called small molecule drugs. So the type of medicine that you can take as a tablet with your breakfast orange juice. So these are well precedented. So when I say that, I mean drugs like this have been brought forward for all sorts of diseases for many years. And we know that drugs like this can work for CNS diseases, so brain diseases and brain degenerative diseases. So for us, it was important to choose a way of treating the disease that we know has worked before for other diseases. We also knew from talking to patients and various other sources that actually people prefer to have a simple tablet that they can take over some of the newer, more complicated ways of treating diseases that might need intrathecal injections or even a stereotaxic surgery for a gene therapy, for example. These are very scientifically elegant ways of treating disease, but they're not the most practical. They're not the one that patients necessarily prefer. Pill you can pop with your
Yeah.
breakfast
I think
is
that's
much simpler.
pretty easy to understand. Actually, I don't know. Would you prefer a pill or a brain surgery?
I fully understand why you would want a tablet that you can take, whether that be daily or whatever the dosage might be, and that it does it does seem a wee bit more attractive, let's be fair. And it's probably, I think, a big thing in the HD community now, and something that's been brought to light more so in recent, months in the past year, is the like the availability, that if one of these
Absolutely.
treatments work, is this going to be available um to everybody around the world? And we're thinking about countries like Latin America, Venezuela, things like that there.
Northern Ireland.
But I was going to say, being from Northern Ireland, even though it's a part of the UK and we do have semi-good healthcare, we don't have access to the likes of this. So it is very attractive to hear that it's in that simpler form of taking a pill, But yeah, it's a bit more attractive, I will say.
The other thing, and I think you make a really good point, Ashley, and this was one of the things in my mind, is that a simple tablet is something that can be distributed around the world. It doesn't
Mhm.
need really complex health infrastructure and, an amazing teaching hospital and research hospital like UCL has to administer it to people. The other thing is, we were well aware that the way this works is that it can slow down or stop the progression of the disease. And so actually, the best time to take it ultimately, these won't be the first clinical trials, but ultimately will be as soon as somebody knows they're gene positive. So long before they've got any symptoms. And there, when you're living a normal life, you've got, busy family, busy day, actually a simple tablet that you can take is really convenient, rather than, for example, having to go, even if it's only to the GP surgery once a month for an injection or something. Actually, the tablet gives you that convenience
Yeah.
that is not there with many of the other kind of newer, more complicated medicines.
Yeah.
And like,
So that's
you go ahead, Ashley.
Sorry. No, I was just gonna say, so like right now, like what we've learned about yourselves is, you seem to have like a huge career in like the medical research field. Like you've been in the HD world for some time, having done your PhD in that. And the goal is to like reduce or lower the Huntington symptoms by giving patients a pill. And we're gonna like deep dive into the science of it and like how we're gonna do that and why we're gonna do that. Um Before we started recording, I did say I had a list because it's I just need to get my head around it. But that's essentially like the elevator pitch top line. That's what you're looking to do as a company.
Absolutely, yes. So our goal is to get a Huntington's a mismatch repair inhibitor, a pill that we think will treat Huntington's disease, into clinical trials as quickly as we can, see if it works in patients, and importantly, it's safe as well, and then get that through the later stage clinical trials, and then available, on the NHS and other healthcare systems around the world for patients' benefits.
So I think we're going all the way to the end point, which I love. Let's get right into the science then. So you mentioned repair. What is that?
What is that? So
Yeah.
this is all about focusing on the DNA in everybody's cells. So I think probably most people listening understand that Huntington's disease is where your Huntington gene has this expansion, more CAG repeats than it should have. And that means you'll go on to develop Huntington's disease at some point in your life. And that actually what kills your brain cells is the toxic Huntington protein. And that's absolutely true. But there's a stage before that. So most Huntington's families, you would inherit a CAG repeat length in your Huntington gene, somewhere between 40 and 50 of these repeats. But actually, that isn't what leads to degeneration in your brain. It's not what causes your brain cells to die. Those repeats actually expand upwards to several hundred, and that's what the problem is. Now, as a scientist, I always thought about the DNA in your cells as being this kind of like tablets of stone, the instructions of how to make you, because you inherited that from your mum and dad. And if you have children, you will pass some of that, half of that information onto your children. So we always think, or I always thought of DNA in your cells as this kind of immutable, solid stuff that just stays there through your life. But actually, it gets a lot of wear and tear. So your DNA in your cells is subject to all sorts of external things that can damage it. So X-rays, sunlight from UV light, chemicals, mutagens, all sorts of things can cause your DNA to have damage. And also it can get wear and tear just by using it. The point of the DNA in each of your cells is that it codes for different proteins that make you work. And so different bits of that DNA are always being kind of read. So cellular machinery kind of unwinds bits of your DNA, reads the sequence, writes it down on a temporary bit of paper called RNA that is then used to kind of transcribe, so to turn it into a protein that is the kind of the nuts and bolts of what makes your cells work. And when it's being, wound up, unwound, etc., it can get damaged. A bit like when you've got your favorite top on, if it's knitted, you can pull a thread and it can all start to unroll, or you might, snag it, or you might spill ketchup down it, which I do quite often. All of these things can kind of damage your clothes. And it's the same way that using your DNA can lead to damage. So because it's really important that your DNA remains, in good nick all the way through your life, there's a whole load of systems in your brain called DNA damage repair pathways that is designed to keep it working well. Lots and lots of different branches of this big kind of system each have a specific role. So some will deal with where a particular base is chemically damaged. Another will be when one strand snaps so you can put it back together again, etc., etc. And we focus on one of these branches, which is called mismatch repair, and that's critically important to the disease processes for Huntington's.
Well, I think I need a new job because David's very good at explaining things simply.
My DNA is a woolly jumper and it's unraveling every time I wear it, i.e. use it. I'm like, he stole my analogies, Lauren.
I know, wait, you don't even need us.
I'm about to hit that end Zoom call button, That was actually a really good way to explain it. So day on day living is using your DNA, whether that be the factors that you mentioned, like sunlight and stuff, or that's just general living. It's wearing away at your DNA. And I'm going to get this wrong, in my body, there is mechanisms and there's things in there to fix that and repair that. And is that what you guys are essentially going after?
It is, yes. So you've got, in each one of your cells, you've essentially got that, hotel sewing kit that if the button's dropped off or you've got a hole, you can kind of sort it out. So there's a complicated set of machinery in there. There's actually involves hundreds of genes that are designed to keep your DNA in good working order all the way from before birth, even all the way through to death. And it's really important that it keeps your DNA sequence exactly as you inherited it, because, at least in the perfect individual, that's exactly how you want it to be. Now, in the case of Huntington's disease, somebody has inherited a gene with this extended CAG repeat, this extended triplet repeat. Now, what we've learned is actually that extended repeat, not only does it cause disease, but it's actually unstable in itself. So because it's too long, what happens is that when it gets unwound and your Huntington gene is then transcribed, and Huntington is expressed, it's present in most cells in the body. So that gene is always being used. It's one of your kind of slightly favorite pair of knickers. It's always being, worn and kept in, washed, put back in the cupboard, used again, or socks if you prefer.
Let's go for the socks.
We'll go with socks. So because it's being used a lot, it's always the two strands of the DNA are being unwound so that one of those is copied to make a new sock, as it were, and then it's wound back up again. Now, when you've got a really long repetitive sequence, it's quite easy for one bit not to get aligned with the other. And to stick with the clothing analogy, if you've got a cardi with a zip on it, it's really important that each tooth so the camera can see, each tooth lines up with the next tooth. What you don't want is kind of two teeth sticking between, or, getting a loop so there's a bit missing in your zip. And this is what happens with a really long repetitive sequence, because it's CAG, CAG, CAG. How do you know that the first CAG is lined up with the first CAG on the next one? Maybe it's kind of slipped a bit and it lines up with the second or the third one. And this is how mismatches that are relevant to Huntington's disease happen. Essentially, you've got two repetitive sequences and they don't quite line up. So you've got some bit sticking out here that shouldn't be, and another bit sticking out there on the other side, so that repetitive bit mismatches. And then, of course, it needs repairing. Now, mismatch repair machinery in your cells should do it properly, keep the sequence the same. But sometimes it goes wrong and it adds extra CAGs in, which is actually making things even worse.
Amazing.
So, does that make sense, Ashley?
Yeah, I know. I feel like
I get this.
We're just gonna go home.
So, if a person has Huntington's disease and has the expanded gene that causes it, all your wee CAGs no longer maybe line up the way they should. So yous are going to be like the dressmakers, like me jumper's overworn, me expanded jeans are overworn, and yous are going to be the people to come in. You're hoping your pill will come in and make sure that they line up again, because the DNA repair isn't working anymore. So yous are going to come in and like try and rejig that to get that going again.
Close, but not quite, Ashley.
Because one thing we didn't really explain is, so David mentioned about the information from genetic studies in HD patients. So these studies were looking for other things in the genome, so the genetic information that everybody has, to see if they affect how early or late someone gets Huntington's despite their standard mutation. So say if you get a genetic test, you find out you've got 41, are your onset to symptoms earlier or later than what we would predict? Because we know that the length of the CAG does really relate on a big scale to how people when people develop symptoms. But we know when you've got 40 repeats individually, we can't predict that, because there can be 20 to 30 years difference of when someone gets the disease. So a lot of the genes that came out of this study, where they were looking across every gene, they kept getting hits that were really significant to either delay onset or speed up progression. So people were earlier than expected, and a lot of these were from the DNA repair pathway.
Okay, so is it going to very much depend where you are in your symptoms if this medication was to work? When a person would start taking it, well, you're not going to reverse what has already progressed, but you'd maybe pause it where it is.
So really good question. So one of the things that I'd always thought about in Huntington's disease is that in the areas of the brain that are degenerating first, so the caudate putamen or the striatum, which is the area that causes those movement, excessive movements and psychiatric symptoms. I'd always thought that neurons in there were basically kind of on a ski slope. They were slowly getting worse and worse over time until eventually individual cells would die and that area of the brain would shrivel up. What we've actually learned, and this is actually very recent stuff that's come out over the last 2 or 3 years, where researchers have used post-mortem Huntington's brain samples and looked at individual cells and through lots of complicated analyses. what they've actually shown is that through the period where your CAG repeat is expanding, actually it's more like a kind of a waterfall kind of thing, where you've got a cell which inherits, say, 41 from Lauren's example, and then it goes up to 42, 43, and then we get into the 50s, and then the 80s, and then the hundreds. And actually, for quite a long time, that brain cell is still, as far as we can see, normally functional, until it reaches a tipping point, the point where on a waterfall you go off the edge, and then there's actually quite a rapid decline for that cell until it then dies. Now, what's really important about this, to your question, Ashley, about when do you treat somebody and when can it be effective, is with a ski slope, you're already kind of halfway down the ski slope if you can stop yourself going any further. With a cliff edge approach, as long as you haven't hit the cliff edge, you can be stopped from going over the end. So all your cells are kind of on this conveyor belt, as it were, getting closer and closer to the cliff edge before they fall off. So what we want to do ideally is start as early as possible, slow that conveyor belt down or stop it altogether. And then those brain cells that would go on to die will never reach that tipping point, never fall off the edge. Now, one of the practical challenges we have when it comes to running clinical trials and proving that this works, because I think it's important to highlight that the human genetic studies have implicated this pathway. Lots of mouse studies and studies by academics in cells in a dish have all supported that this should work. But ultimately, the proof of the pudding is for us to take either a LoQus23 medicine or one of the many other companies working in this area into clinical trials and show it actually does what it says on the tin. So what we will need to do is show that we can slow down the progression of Huntington's disease. Now, where we will probably start for practical reasons, the easiest way of showing you can slow something down is if you have a patient who's got symptoms, you can measure those symptoms and then look over, say, a year or 2 years of treating with your drug and say, have those symptoms got any worse? And that is much easier to do than taking somebody who's in that pre-symptomatic stage and saying, have we slowed the disease down? Because there are things we can measure there, but they're harder work to prove that you've absolutely affected the disease. So for good scientific reasons, and this is what the drug regulators will want to see, is that we can slow down symptom progression in the first instance. As soon as we know that works, then you've got a much stronger case to go to patients who are not yet symptomatic. So let's say we predict your symptoms would start at 45, say. Then actually what we want to be treating is those patients who are 35 to 45 and stop them ever getting those symptoms, or at least make it take a lot longer. Mm-hmm. Now, why do we think that this will work? And it comes back to what Lauren said earlier. For somebody who inherits, say, 41 repeats or any repeat length, we can make an estimate based on averages with people with that repeat length of when the disease starts. But there's an enormous variation. Could be 20 years earlier, could be 20 years later. So what we want our drug to do is essentially push you to that 20 years later or 30 years later point, so that you either have a lot longer of healthy, unimpaired lifespan and really, quality of life, or, best case scenario, it never happens. So this will be something we've got to go stepwise, I'm afraid. But, we've got, I think, a really good chance of being able to do it now.
So basically, going back to kind of where you are at with your program. So you've had a big announcement recently. I know mainly you've been what we call preclinical, and you've talked a lot about that. So preclinical just means when it's still on the lab bench, kind of really scientific testing stage, there's quite a long period of this, I think, in drug development before you can even approach the clinic. Do you want to tell us what the announcement was, what it means?
Yeah, sure, Lauren. So the key thing we announced at the beginning of October is raising what we call a Series A. So it's basically the next lump of money that we've raised from hang on, just Give me a second to stop something that's going to make lots of noise. Right, so we we have a great messaging system. Someone's telling me about some science, but at the moment I just want to concentrate on talking here. So raise a lot of money that allows us to take from where we are now, which is, as Lauren says, preclinically. So we're doing tests in cells and in Huntington's transgenic mice, and then take that through to, first of all, safety tests, make sure it's safe, which is critically important, and then into what are called phase 1 trials, where we first give it to people. And the first thing we need to do is check it's safe, and it it kind of does what we expect. So when you swallow the pill, some of the drug ends up in your brain, and then we can go on to say, well, okay, now we know it's in the right place and it's safe. Does it work in patients? So the reason it takes us a long time to do this bit is we've got to find just the right molecule that will hit our particular mismatch repair drug target really, really selectively, doesn't hit anything else, because the thing about DNA damage repair systems is when they go wrong, yes, they can cause Huntington's disease and other neurodegenerative diseases, but they can also do things like cause cancer. So we need to make sure that we're very selective and we just pick out the little bit of that massive machinery that is going to affect Huntington's disease and not do anything else bad. Now, it turns out that the target we're going after, which is called MSH3, and is really hard work to find a small molecule for. I've worked on many different drug projects. It's got to be right up there of difficult to do, which is why it's taking us quite a long time. But we've got some really, really nice data, and we're getting close to choosing what we call a development candidate. Now, one molecule that you pick, and then you carry that forward into your clinical trials. And picking that one molecule is very important because you've got to live with your choices. It's a bit like voting for a president or a prime minister, right? It only happens so often. But when you pick that one molecule, you have to live with those choices. So you want to be very careful which one you pick. So that's the bit we're working through. We're still campaigning, as it were, to make that one key choice.
If you pick the wrong molecule, I'm thinking like I'm getting dressed on Saturday night to go out and I visualize an outfit in my head. We're very related to fashion in this talk. I picked an outfit in my head and I picked a pair of shoes. No, they look absolutely awful. Can I change my shoes? Can you change your molecule?
You can, but if you only realize that you've got, the wrong shoes on when you get to the restaurant, the only way you can change your shoes is you've got to go back home, root around in your wardrobe, pick out a new pair, and then go back out again. So you can do it, but it means you're going to be half an hour late for dinner. And that's the problem that we will have. If we pick the wrong one, and it can be wrong for all sorts of different reasons, it might not be safe enough. It might, when you swallow it, it just comes straight out the other end, never gets absorbed. Lots and lots of things can be wrong with it. Then we have to go back and pick another one. Now, one benefit is that we learn from that process of what was wrong with the first one. So when we pick the second one, if you pick the red shoes and it didn't go with your pink outfit, you don't go home and pick another pair of red shoes. You then pick a different color. So you learn, but of course it takes time. And the thing that patients don't want is to be sitting around waiting for therapies to come along. So it's important, and we are trying to make sure we pick as best as we possibly can, so that when it goes to the clinical trials, it does what it says on the tin and then can go through to patients.
So is there a deadline to make this big decision, or when's election day?
So definitely my investors are very keen to know when election day is. So we're at that stage where we've got a lot of preclinical data. We know it works in biochemical assays. It works in cells. We're just testing it in mice at the moment. So next year we plan to pick that one molecule, and then we have to do
2026 next year.
Sorry?
2026 next year. We're very close to
2025 next year, 2025. So pick that one molecule in 2025. Then we have to make, a big bucket full of it, test it in safety testing to make sure it's safe to give to people. And then in 2026, we should start giving it to people, seeing if it does what we hope it will do.
Exciting.
That's interesting.
I think you've just
You say it out like that. Like it's a very complex thing you're trying to do, and you're obviously you have to be very careful, but 2026, like as much as, like I live at risk of the disease and stuff. So it's, I'm definitely one of those people that's like looking at my watch going, right, you done yet? I'd be more annoying than your investor. But at the same time, it's like, okay, so you could potentially be on human trials by 2026.
Yeah. Yeah.
Can
And
I
the
ask,
other
do you know where
important
they're
thing
going to
actually
be?
is this science approach is one that a number of different companies are taking, not just LoQus23. So, from your perspective, you don't just get to buy one lottery ticket. Actually, you get to buy, 2 or 3 strips worth, and any one of those could be a winner in terms of showing it really works in Huntington's patients and slows the disease down. And then, hopefully, I hope that ours works because we think it's the most convenient way for patients to take their medicine. But, if ours doesn't work for some reason, it falls over because of a safety risk or something else that we didn't anticipate, then if a different medicine that's also against the same hypothesis going after the same target, but is maybe, an injection or something else, well, that's still good for patients because that gives an alternative way of treating the disease.
Absolutely.
And of course, if several of us
You think
work,
you've
then that
answered
also means
questions.
that you can choose, as to what's most appropriate.
Yeah.
Yeah, it's hard to ask you more questions because you always answer very well. And
I was
Well,
going to be like, are
what?
you going to do
I
if the
think
pill
it's.
doesn't work? Like, are you
Oh,
going to do one of the more like invasive routes to get this thing into my brain? You keep
but you already
answering.
answered it.
one of the other big advantages, we talked about the advantage when a medicine's available on the NHS or healthcare system, but one of the other big advantages of a small molecule, as we call them, so a pill you can just swallow, is that for perhaps people like yourself, Ashley, who would want to be part of a clinical trial to see if it works, it's much easier to go broadly out to different medical centres who can then run those clinical trials because they don't need fancy equipment or complicated ways of dosing a drug. So it means that we can go, hopefully, to medical centres where the patients are, rather than trying to get the patients to come to the medical centres that can technically manage what we do. And I think that is helpful for us to be able to go to places and recruit people more quickly, see if it works. And hopefully for people listening to this video that, who don't live next to UCL or within spitting distance of Mass General in Boston, they actually live somewhere, perhaps with a nice view, like Northern Ireland. And so then, it's easier to enroll in those clinical trials.
That's the hope.
That I
And
mean, I think
that gives
that's
us 2 years to get enrolled in Northern Ireland, actually.
we're working on it. I hear we're working on it. No, I suppose that is quite good. And like I know a lot of like my friends in the HD community will like to hear that because there is so many of us that would love to get involved with a clinical trial. And I know you can sometimes travel to do clinical trials and stuff, but it's hard because even if you get compensation, you're still taking the time off work. And we're not big science head people. So, if you tell me I'm going to be okay, it doesn't mean that my brain's going to register that. I'm still going to be scared that, like, what if something happens to me and I don't have, like, Professor Wild next door in the hospital? I'm all the way over the pond. So it's definitely nice to hear that it's something that can be given like you might venture further because I fully understand how hard it is to set up clinical sites and I fully understand why they are where they are and stuff. Like I'm not arguing that. I'm not trying to, come down hard on anybody else. But it is nice to think that it might venture to places that don't have anything and give that opportunity to other people who haven't, been fit to get involved. But as a whole, like as somebody who can't get involved with clinical trials because there's nothing in my country. I am very grateful for those that who do and are able to and that, put themselves forward for that. But I'm sure a pill's probably quite attractive to them, to be fair. Um, it seems a little easier on you. Um,
So where can the community find out more and keep up to date and maybe hope to find out who gets elected next year?
Yeah.
So we do have a website, LoQus23.com. Now, it doesn't have, I'll be honest, it doesn't have a huge amount of information on there at the moment. Now, as we select that development candidate, then we'll be putting more information out there about what we're doing, but importantly, talking to the community so that people know that we're coming along. And so, come 2026, people know who we are, know what we're doing, why we're doing it, how it compares to other options, because we're well aware that by the time we get to running clinical trials in Huntington's disease, there's probably quite a lot of companies out there with different medicines, some of which will work the same way that ours does, others will work in different ways. So we think it's important that people understand what we're doing, that then, depending on where you live and which trials are running out of which medical centres, you as patients can make, a really informed choice of, would you like, obviously with the help of your doctor, should you go on this trial or that trial, or wait until you see some results. So myself, Caroline, other members of the team will certainly be doing more of things like this, talking to people, explaining the science that we do. And then we'll also be going to more meetings, congresses, etc., so that we can talk directly to patients and their families, hopefully answer questions in clear ways, and hope that one day we can recruit you guys into a trial and see if our medicine works.
And
So
for anyone who doesn't,
PR.
for anyone who would like to a refresher on the kind of science about what we were talking through here, it's called somatic expansion or somatic instability and DNA repair. We have another breaking down barriers video that's on our YouTube that was done with HDBuzz, and goes into that a lot more if you're interested in kind of understanding the science a bit more. And I obviously have to do a plug for the HDYO Congress, which is in Prague next year, March. I'm going to lose the date.
St. Patrick's weekend.
I know it's St. Patrick's weekend, so around the 17th of March.
14th, 15th, 16th of March. I'm waiting for Jenna to pop up on the chat being like, no.
Anyway, it's on our website, and it's going to be linked, I'm sure, to this video.
And we got it right, guys.
We got it right. We're all good. 14th to the 16th. So I think that's all we have time for now.
Lauren, one more thing.
Oh, yeah, go ahead.
We need to cordially invite David to Congress so that we can all go and talk to him about our woolly sweaters.
I love it. I mean, I think we might have David come do some science sessions because he's obviously good at explaining complex things. So I hope Jenna notes this.
I'm going to get kicked out of my job.
Can I put one plug in? I gave a presentation at the HDSA. It was one during COVID, so it was all done virtually. And there we used cuddly toys and some tape and a whole load of photographs to try and explain what mismatch repair is. So if you look at the news page on LoQus23, you'll see somewhere a little bit down the list that we presented at HDSA. If you click on that, it'll take you through to a video. I think you have to log into the system there called Vimeo, but then you can watch the video. So there's some boring PowerPoint slides at the beginning and at the end, and then there's some fun pictures while I try and explain what's going on.
And Jenna will probably add links to this video, so you can be sure to catch that. So as we say goodbye, any closing thoughts?
I just want to say, the thing that makes it possible for LoQus to do what we do is patients contributing their time, their blood samples for genetic studies, sometimes their brains after they've passed away, that allow us to understand how Huntington's disease works. And that's what gives us scientists the insights of how we might try and treat this. And then ultimately, we need your engagement for clinical trials to then see if our ideas are right and they work and can produce really effective medicines. And I know this is, when I did my PhD, there were no effective therapies that could slow down or stop the progression of Huntington's disease. Sadly, that's still true today, but hopefully, you know, in the next decade or so, we can solve that problem. So thank you, everybody.
Thank you. That's a nice way to end. I don't know if you have any thoughts, Ashley, but
it was a really lovely ending, and thank you so much. You explained it in a really good way. So I really appreciate, you know, helping me and everybody else to understand who doesn't come from a science background. So I really do appreciate that. And I was being serious about the invite to Prague. So I'll see you there. With a woolly sweater because I Googled it and it's really cold over there in March. So wear your woolly sweater.
All right,
amazing.
noted.
All right. Thanks,
Thanks, Ashley.
everybody. We'll see you at the
Thanks.
next video.